Today we will talk about another signaling pathway which is very important in cancer. I will tell you why it is very important. All of you are familiar with what is called apoptosis. Some people pronounce it as apoptosis also. The P is silent, I believe. It is a programmed cell death. It has significance not only in cancer, but in many physiological processes. For example, I will show you some examples where you will be able to understand it better. Look at this picture, especially this one. All of you know when during animal development, in the initial stages, when fingers develop, there are what are known as interdigital tissues. You see the red ones there in between the digits, because it is animal, they are calling it as digits. So, interdigital tissues. When the fingers have to emerge, when the digits have to emerge, these interdigital tissues have to be destroyed. That is done through apoptosis. Apoptosis is a form of programmed cell death, I told you. So, this death is very essential during development. Not only that, another famous example is the development of the frog during the so called metamorphosis. The frog, adult frog has a tail, but I mean like adult frog, ultimately the tail disappears from the adult frog. When it is in the early stages, you have a tail, right, what is called something like a toad or something. So, during metamorphosis, the tail disappears. Again, that tail tissue disappears due to apoptosis. So, apoptosis has many roles. Not only that, during our own colon formation, colon, it is like a tube like structure. Initially during development, it develops like a rod. You know the difference between rod and tube, right? Rod is just a heavy stuff. In between there is nothing, it is a hard stuff, right? So, the in between that tissue should be destroyed, so that you form a canal like structure. So, that is also done through apoptosis. And some of the vestigial structures during the reproductive organs, there also the Mullerian duct, Wolffian duct, for their development also you need this apoptosis. Not only that, in some organisms there must be some set number of cells, 1000 cells or 2000 cells, something like that, for that organism. But during the initial stages of development, the cell number will be more. So, you have to reduce that cell number by eliminating the unwanted cells. So, that is also done through apoptosis. The story does not end there. When you have some infection also, the infected cells have to be destroyed. So, that also is done through apoptosis. So, that means in the immune system. So, apoptosis has several roles in our physiology. So, that is the importance. Helps in the organism eliminating unwanted cells with minimal damage to the surrounding cells. An organized process in which various cellular components are broken down systematically. We will look at it how that is done. Now, since we are talking about cancer biology, we will talk about its relevance to cancer. Every cell in a multicellular organism has the potential to die by apoptosis, because you see, if it is a single cellular organism, unicellular organism, if it commits suicide, what we call as a suicide for apoptosis, that is because in suicide the person who wants to die decides that he wants to die, whereas in a murder somebody else decides that he or she has to die, right. That is akin to what we see in necrosis, that is another form of cell death. where it is not those cells which are dying, they decide, they do not decide. Some other force, maybe a strong force, maybe a knife, maybe some fire, maybe some heat, that destroys that. Whereas here it is not. Some signals come to the cell and the cell decides to die. So, that is why we say that as akin to suicide. Of course, these examples are given just to make you understand. You cannot take it like for every example whether it will be suitable and all that. Do not take it that way. The tumor cells, they evade apoptosis, they refuse to die because they want to live, they want to propagate, they want to proliferate. So, they have developed certain mechanisms, faulty apoptotic pathways. So, the apoptotic pathways in the normal cells are different from the tumor cells. Tumor cells have faulty apoptotic pathways. For example, if you damage the cells with radiation, the DNA will get damaged. The cells will, normal cells will try to repair that. If they cannot repair, they will undergo apoptosis. That is, if a particular threshold is exceeded, then they will commit suicide or they will commit this apoptosis, they will enter into apoptosis. Whereas tumor cells, you will not be able to do that because they have faulty apoptotic pathways. So, they will refuse to die. So, this is how but although I am saying in a very broad sense, there are specific mechanisms by which they are able to evade apoptosis. There are specific molecules which are mutated or altered in the apoptotic signaling pathway, which we will systematically study. So, what is the consequence of this? These defects not only increase the tumor mass, that is tumor grows bigger, not only that, but also gives them an advantage for growth, and so the tumor cells become resistant to therapy. Now comes the importance, right. The cancer cells become resistant to therapy if they have certain anti-apoptotic molecules being expressed in excess. So, they refuse to die due to treatment. Why we are talking about treatment is because this is the most relevant point about apoptosis. Most of the anti-cancer agents kill the cancer cells through apoptosis. So, in other words, apoptosis appears to be the major mechanisms by which many of the or most of the anti-cancer agents act. So, if you can devise something to induce apoptosis by anti-cancer agents to cancer cells, but not to normal cells, you got it. So, that will be one of the best treatment methods. So, that is why people are interested in apoptosis. So, I think I have given you an overall idea why apoptosis is important in cancer. That is, many anti-cancer agents act through apoptosis. So, if you can devise a method by which these anti-cancer agents can induce apoptosis in cancer cells, but not normal cells, that will be a very good therapeutic strategy. And cancer cells have developed several mechanisms by which they will have resistance to apoptosis induced by any agent, any chemical. Because they have faulty apoptotic pathways, they have developed certain defects in the apoptotic pathways, or alternatively they are over expressing anti-apoptotic proteins, and thereby they can resist apoptosis induction by any agent. This field is very important because you see Nobel Prize in Physiology and Medicine in 2002 was awarded to 3 scientists. One is Sidney Brenner. He established C. elegans as a C. elegans, you know. So, it is a what? Nematode, it is a nematode, right. So, as a novel experimental model organism, incidentally you see for receiving Nobel Prize in medicine, people have worked in lower organisms, right, because the knowledge gained through lower organisms have always enriched us with so much ideas that we are able to develop modern treatment methodologies. The knowledge gained through them, oncogenes, tumor suppressor genes, many things can be tested. So, these models are very helpful. Similarly, Robert Horwitz, he also characterized key genes controlling programmed cell death in C. elegans. And of course, these genes, because he discovered that in C. elegans, later on people found that the very same genes, homologs, are present in humans also. So, that was made use of by John Sulston, who showed that specific cells undergo programmed cell death as a part of normal differentiation process, and he identified the first mutation of a gene participating in the cell death process. So, these are the important discoveries. Let us look at the apoptotic cells. I always used to, because we also do some work on apoptosis in cancer cells, I tell my students usually, before you do any experiment, just look at the cells through microscope, just to see whether they are undergoing apoptosis, whether cells are dying. Suppose you want to understand whether a particular Ayurvedic concoction or some plant extract has anti-cancer property, the first thing you will do is you will add that extract to cancer cells in the lab and then look at whether they are dying, right, or you will increase the concentrations and see if it is concoction, you are going to increase the amount of that concoction that you are adding in terms of, you will measure it in terms of dry weight or some volume, whatever you will progressively increase that and then see whether there is a progressive increase in the cell death. So, in order to look at that, you should look at the cells through the microscope. So, the normal cells look like this flattened structure. You see the apoptotic cells, they develop certain blubs, certain bulgings, and then some of them develop spike like structures, and finally, you also see some blisters on the cells. These are some typical features that you find for apoptotic cells in culture. I will come back to that structural details a little bit later. Before that, apoptosis is not only important in cancer, but in several diseases. With respect to cancer, you want chemicals that will induce apoptosis, but there are other conditions, diseases, where there is already increased apoptosis. For example, neurodegeneration, where the cells are undergoing apoptosis. So, you want to stop that. So, if you discover chemicals from any source that induce apoptosis, they will be useful in cancer probably. Of course, you have to do so many tests before these chemicals can be qualified as anti-cancer agents. At least they will have the potential, right, which needs to be tested. Similarly, if you have chemicals that inhibit apoptosis, they are also useful in some other conditions. For example, in neurodegenerative disorders, there is increased apoptosis, so you want to reduce that. So, that is the importance of this slide. So, AIDS, neurodegenerative disorders such as Alzheimer's, Parkinson, retinitis pigmentosa and so on, myeloblastic syndromes, ischemic injury, myocardial infarction, toxin induced liver disease like alcohol related liver disorders and so on. So, these cells are dying, there is increased apoptosis. Inhibition of apoptosis, obviously we are interested in cancer. In cancer there is faulty apoptotic pathway, so there is inhibited apoptosis. So, in this condition, so you want chemicals that can induce apoptosis, that has to be kept in mind. So, there are many cancers, carcinomas with p53 mutations, hormone dependent tumors, breast cancer, prostate cancer, ovarian cancer, follicular lymphomas. In all these cancers, cancer patients have been found to have some defects or other in apoptotic signaling pathways. Most of them either have defects in the pro-apoptotic gene or have increased expression of anti-apoptotic genes. In fact, they have both. Many cancer patients have defects in both. Pro-apoptotic genes will be mutated or they would have lost the expression because they are tumor suppressor genes. So, there will be loss of expression of that, deletion or mutation or whatever. Simple thing is inactivation. Inactivation of tumor suppressor genes, activation of the oncogenes. In this case, it is anti-apoptotic genes. I told you anti-apoptotic genes are more or less like oncogenes. So, autoimmune disorders and then in certain viral infections, you have inhibition of apoptosis. I think you should not get confused with the inducers of apoptosis with this. In these conditions, there is increased apoptosis or decreased apoptosis. So, that you have to understand. Like I said, we will come back to the structural features. Some of the biochemical features are chromatin is very much condensed in apoptotic cells. There is a fragmentation of DNA and the phosphatidylserine which is usually present on the membranes, it gets externalized. We will look at that why it is important, I will just tell you. And then one of the caspase 3 substrates is called PARP. The PARP is cleaved, that means caspase is activated. Cytosol shrinks and there are vacuoles. You see so many vacuoles in the cell, apoptotic cell, and you also see apoptotic bodies. The vacuoles are shown here. So, you start seeing many vacuoles. So, that is one of the symptoms when you look at the cells through the microscope. So, that is one. Mainly you look for so called apoptotic bodies. That is what we have to do as the first test whether the cells are dying. These are some of the sequence of changes that occur when a normal cell becomes an apoptotic cell. So, first thing is cells shrinkage away from neighboring cells. So, that you see the nucleus is coming to one side and then as I told you some blubbing and cytoplasmic nuclear condensation happens, margination of the condensed chromatin, that is what I told you, it goes to one end, margination of the condensed chromatin and slowly DNA fragmentation, nuclear and cellular fragmentation, and it forms apoptotic bodies. What is interesting is that the cells are not burst out, but form certain apoptotic bodies, they have their own membranes. So, they are intact. So, that is a characteristic feature. Finally, of course, cells have to die and through phagocytosis. That happens in the immune system in many systems. As we go along, we will discuss that. Now, this slide also shows you some of the important differences between the two forms of cell death. Upper portion, upper panel is depicts the events occurring in necrosis. The lower panel is depicting apoptosis. So, you can see that what I just told you, in the apoptosis you do see DNA fragments and all that, but you see the intact organelles, apoptotic bodies, right, intact. The membrane is not totally disrupted in the initial stages. Whereas, here you will see the cellular content is almost, the membrane is broken down and the cellular content is almost vomited out. So, the chromatin pattern is kind of conserved, mitochondrial morphological changes do happen, reversible swelling happens as opposed to condensation here. So, these are some of the differences, but I have also tabulated the differences. The main difference is there is no inflammation during apoptosis, whereas in necrosis the cells become inflamed. So, that is one of the important things. I have just for your understanding, I have shown you some of the differences, major differences between apoptosis and necrosis. You can see that the nuclei in apoptosis, there is a dense condensation of chromatin, whereas irregular chromatin clumping is there. Cytoplasmic organelles are intact in apoptosis, whereas they are disrupted in necrosis. Cell membrane, the blubbing and loss of integrity, more or less you see that. Cells shrink and cells swell. This is another important difference. Single cells are affected because each cell decides based on the signals it receives whether to die or not. So, it is a single cell can be affected, whereas here it is group of cells. Inflammation is observed in necrosis, but no inflammation in apoptosis. So, these are the major differences that you may have to understand. Now, you may be wondering how this is all happening. The major player in the execution of this cell death is called a caspase. Caspase is an enzyme. Caspases are nothing but cysteine proteases. They cleave aspartate in the first position of tetrameric recognition sequence. They have a recognition sequence. The first amino acid is aspartate. So, usually these caspases are synthesized as like other enzymes. You have heard of for example, trypsin, you know how it is synthesized initially as inactive form and then it becomes active form. Similarly, here also inactive zymogens as they are called. The enzymes, inactive forms of enzymes are called zymogens. So, they are activated by this aspartate cleavage. So, they remove the N terminal pro domain and thereby they become active forms. So, this is one mechanism. As usual in biology, you have so many substitutes and caspase does not mean it is involved in apoptosis. There are number of caspases, as many as 14 mammalian caspases have been identified. Not all of them are involved in apoptosis, right, but many of them are involved. 11, 1, 11, 4 and 5 are non-apoptotic. They are not participating in apoptosis. About 10 caspases are implicated in apoptosis. The main mechanisms by which they are activated include what is called auto activation. That means the caspase 8, procaspase 8 activates by let us say phosphorylation, for instance. It can phosphorylate and then it can get activated. So, it activates itself, that is auto activation. Trans activation, that means one caspase activates another caspase. So, 3, 6 and 7 can be activated like that. And finally, there can be a simple conformational change. So, there are basically about 3 mechanisms by which caspases can be activated. Usually, they are in the inactive form and they are activated by that proteolytic cleavage and they can be activated by trans activation, like one caspase can activate another and then through conformational change. So, these are the mechanisms. Now, we will look at why okay, here originally when they were discovered, they were called interleukin converting enzyme family as cytokine processors during the initial discoveries. So, they were called ICE family of enzymes, that is interleukin converting enzyme, that is how they were called. So, the family of caspases were known as ICE family, that was the original name. Again, you had homologs in the lower organisms like ICH, NED2 family of apoptotic initiators in the C. elegans and other organisms. That is how see SED family, C. elegans. Now, I am coming to another important question. How these caspases are able to induce cell death? The cells are held together. There are 2-3 important things. For the cell, what is important? DNA is very important, nucleus. So, DNA fragmentation. Once the caspases are activated, they will activate certain DNases. They are known as CAD, caspase activated DNAs. As usual in biology, if you have an activator, there will be an inhibitor. I told you. Similarly, CADs will have ICAD, that is inhibitor of caspase activated DNases. So, CAD ICAD system. In this manner, they can fragment the DNA. So, how will you dismantle the cellular machinery in a systematic way? The first thing is attack the DNA, disintegrate it, so that the information does not pass to the other cells, the cells have to die. The other thing is how the cells are held together by, if I am standing like this, it is because of the backbone, right. If the backbone is broken, there will be a bending. So, break the backbone to dismantle. Cytoskeleton. Cytoskeleton proteins are the substrates. You can see that structural proteins, lamin A, B, G-actin, fodrin, gelsolin, all these are substrates. By understanding the substrates of caspases, you can understand the mechanism by which they ultimately dismantle the cellular structure. Cells are very well organized. So, you have to disorganize that to kill them. So, this is what is happening. At the same time, this killing is a little bit physiological kind of thing, because you can also make use of them again. That is when we discuss the autophagic mechanisms, I will tell you. Once they are broken down, some of the compounds can be taken up again also, but let us not confuse that here. And then signaling proteins. Many of the signaling proteins are substrates for caspases. 3, 6 and 7, they are very important. You can see here that RAS-GAD, then PKC, FAC, these are kinases, PAC2, RIP and so on. There are several kinase molecules. Cell cycle proteins, DNA repair, later on I will talk about DNA repair, PARP, PRB, retinoblastoma, MDM2. MDM2 is anti-apoptotic. So, cleaving that will help to make it apoptotic. So, DNA protein kinase, some of the anti-apoptotic proteins also, like BCL2, BCLXL and so on. Strangely, some pro-apoptotic proteins also can be substrates like BID, and then the nucleases I told you just now, caspase activated DNAs and inhibitor of caspase activated DNAs. So, these are the important substrates. So, caspase is very important in the process of apoptosis. That is what we understand, right. Now, what induces apoptosis is very important. Naturally, I already told you many anti-cancer agents induce apoptosis. So, you can see that the chemotherapeutic drugs such as cisplatin, doxorubicin, bleomycin, cytosine arabinoside, nitrogen mustard, methotrexate, vincristine, these are all very well known chemotherapeutic drugs. Again, radiation can be used to treat cancer, right. It is also an agent, therapeutic agent. So, UV radiation. So, all these things, those which are used in therapy, they can induce apoptosis. There are other damage related activators, that is heat shock, viral infection, bacterial toxins. If you remember, even oncogenes, if overexpressed, can induce apoptosis. I already told you about this, if you remember. Oncogenes are growth promoting genes. Normally, when you transfect them, you expect a cellular proliferation. Yes, that is correct, provided they have some cooperative oncogenes. I also told you about that. For a normal cell to be transformed into a cancer cell, It is not that one oncogene, you need the cooperation of 2-3 oncogenes so that they will be ultimately converted into cancer cells. I told you about this. However, when you overexpress a particular oncogene, it will actually induce apoptosis, which it is not supposed to do, right. It is supposed to promote growth. Here it is inhibiting growth, rather it becomes cytotoxic and it kills the cells. So, you have to understand that. Then some tumor suppressor genes like p53 are very well known to induce apoptosis. In fact, one of the major functions of p53 is to induce apoptosis. Cytotoxic cytolytic T cells, free radicals. Another important one is nutrient deprivation. If you withdraw all the growth factors, then also it can induce apoptosis in cells, growth factor withdrawal for example, nutrient deprivation and so on. Physiological activators are usually tumor necrosis factor, we will study about that, so called TNF, Fas ligand, TGF beta, glutamate, dopamine, N-methyl D-aspartate, growth factor withdrawal I already told you. Loss of matrix attachment, because the many cells when they grow they need an attachment. If you remove that attachment, they will die also, but that is usually known as anarchis. But in certain if you remove the matrix, then it can also undergo apoptosis. Calcium, glucocorticoids and so on. There are many inducers that have been identified. In fact, people are interested to know more about inducers of apoptosis, because such inducers have the potential to be used in cancer therapy in one way or other. So, people are interested in that. Now, how it is done as a signaling pathway, this is a very important thing. I told you signals cannot be defined into one category. Signals can be ion, calcium, it can be growth factor, it can be it can be of many categories. Now, here we are talking about death pathway. So, there are what are known as death ligands. The signal can come from outside or inside. If the signal comes from outside, it is called extrinsic pathway. If the signal comes from inside, then it is called intrinsic pathway. So, there are two main pathways of caspase activation. I told you caspase has to be activated for apoptosis, right. So, there are two ways. The most famous example of extrinsic pathway is the tumor necrosis factor and its related proteins, Fas also, Fas ligand. These are the two important examples. So, you have a death ligand and they have to bind to a death receptor. So, there are also many death receptors and then the signal passes through several caspases. In this case, there is what is called caspase 8. Sometimes they are called initiator caspases. So, you have signals, you have signal receivers and then there are what are known as caspase activators. This is known as TNF receptor associated death domain, TNF receptor associated death domain, we call it as TRAD, we abbreviate it as TRAD. So, that has to be activated by this binding of the TNF to its receptor for example. So, then it activates what is called caspase 8, which is called an initiator caspase. Finally, that activates caspase 3 and caspase 3 is the final executor. So, that is why it is called effector caspase, sometimes called executioner caspase, something like that. So, the people call that way. Caspase 3, 6 and 7 fall under that category. This is what it constitutes, what is called extrinsic pathway. Now, you have intrinsic pathway, signal comes from inside, that is you have survival factor withdrawal, some of the developmental signals, chemotherapeutic drugs also you are giving, you may inject it or something, but ultimately they go inside and do that. That is what it means. What it means is that developmental signal, mitochondrial pore opening. One of the most important events in the intrinsic pathway is the mitochondria open up and leak certain molecules that initiate cell death. One of the most important molecules that is getting released from mitochondria, several molecules are released. One of the most important ones is cytochrome C. You remember cytochrome C, traditionally mitochondria are known as powerhouses of the cell, and cytochrome C also is known to be an important molecule in respiration. Why we are calling it as powerhouse? Because you get the energy ATP, respiration and all that. So, mitochondria are traditionally known for that, but somewhere in if I remember correctly, somewhere in the 1990s, cytochrome C was also found to be important for cell death process. So, it was a very late discovery. Traditionally, mitochondria are known for this energy production, and they are typically known as powerhouses of the cell. But cytochrome C again is an important component of that respiratory process, but it is also important for cell death process. So, cytochrome C is released, and that combines with another molecule called apoptosis protease activating factor 1, APOF1 it is called. It forms what is called apoptosome complex together with caspase 9. Caspase 9 gets activated as against caspase 8 in extrinsic pathway. In the intrinsic pathway, it is caspase 9 which is getting activated. Ultimately, the junction is caspase 3. So, you can see both the extrinsic pathway and the intrinsic pathway converge on caspase 3. This is the important point. So, you have a convergence. Another interesting feature in this is that these two signaling pathways, you do not have to think them as separate ones, they can be connected also. You can see the connecting point from caspase 8. It cleaves a molecule called BID. Once it is cleaved, the cleaved molecule is called truncated BID because it cuts it. So, it becomes truncated. It is called a T-BID. So, caspase 8 can cleave BID. BID is a protein and it becomes truncated. So, it is called T-BID, truncated BID, and that is an inducer of cytochrome C release from the mitochondria. So, this is a connecting point. So, these act as a circuit, right. There are these are not operating individually, they are also connected, they are well connected with each other. So far we have seen that this is how cells are killed, and once caspases are activated, cells can be killed by those mechanisms that I told you, because their substrates are substrates for DNases, for example. So, they can disintegrate DNA, they can disintegrate the cytoskeleton, they can activate several signaling pathways because the kinases are there, they can inactivate also. So, in this manner they will slowly disintegrate this whole cellular machinery and the cells die. So, this is the mechanism. So, you understand that caspases are very important. Then the question comes, some researchers have thought, is it that always you require caspases? Is it that apoptosis will happen only in the presence of caspases? Is there anything called caspase independent apoptosis? So, people have done research on that. What do you think will be the answer? Yes or no? Yes. There are what are known as, so the question is whether the apoptosis is always caspase dependent or independent. There are number of molecules, you remember, that are leaking out of the mitochondria during apoptosis. There are many of them. Some of the pro-apoptotic molecules are called, see the BID I told you. The other important pro-apoptotic molecules are called BAX and BAC. They what is called oligomerization. They form oligomers. They act like pores. And this oligomerization is blocked by anti-apoptotic proteins. The first anti-apoptotic gene discovered is BCL2, because it is BCL, because B cell lymphoma. It was discovered in B cell lymphoma. So, that is one of the first anti-apoptotic genes discovered. The intrinsic pathway, mainly it is called intrinsic pathway because of the involvement of mitochondria and the leakage of certain proteins that come out of mitochondria, and they initiate the process. So, that is very important. Number of proteins come out of mitochondria out of some signals, it can be developmental signals and so on. One of the proteins that come out of mitochondria is called endonuclease G, you can look at it. This can do DNA degradation without the involvement of caspases. That is why people think that it can be caspase independent. Similarly, reactive oxygen species can oxidize the lipid components in the membrane and other components of the cell, and that can also initiate ultimately cell death. There is one more factor which was discovered in France, apoptosis inducing factor. This is also said to initiate apoptosis without the involvement of caspases. It initiates DNA degradation. So, basically at least 3 factors are known to be acting independent of caspases to initiate apoptosis: ROS, reactive oxygen species, endonuclease G, and apoptosis inducing factor. Now, apart from those which we have studied yesterday, there are other number of other molecules. One such molecule that comes out of the mitochondria is called SMAC. It is secondary mitochondrial activator of caspases. So, that is another thing, and there is one more molecule called OMI, OMI. What they do is they inhibit what are called inhibitors of apoptosis proteins. I have repeatedly mentioned that if you have an inducer, there will be an inhibitor. If you have an activator, there will be an inhibitor. So, every time there is a pair of molecules which will act in an opposing manner. So, as the name implies, these inhibitors of apoptosis proteins, IAP, again you have many of them. I told you always, this is another concept, always you have some substitute molecules, IAP1 and IAP2 are there. So, these IAPs are inhibitors of apoptosis. If you inhibit these inhibitors, you are stimulating apoptosis. So, essentially SMAC and OMI, they induce apoptosis by inhibiting these inhibitors of apoptosis proteins. Okay, you may ask what these IAPs inhibit? They inhibit caspase 3 and caspase 9. So, if you inhibit the inhibitor of caspases, then actually you activate the caspases. So, that is what happens. So, this is a very important mechanism. Apart from that, what you need to know is that there is a whole set of BCL2 family of proteins which can act as pro-apoptotic factors and as well as anti-apoptotic factors. Shortly, I will be discussing that. But a pair of proteins, you must remember, Bax and Bac, which form an oligomer which can act as a channel for these proteins to come out also. This BID is another protein. If you remember, I told you caspase 8 can cleave BID, and then that can result in what is called truncated BID, which is a connecting point between extrinsic pathway and intrinsic pathway. These points you may have to kind of remember.